Welcome to today's lunch symposium, The Ongoing Evolution of Co-Stimulation Blockade in Organ Transplantation. This is a symposium that is supported by Veloxis, but the planning committee actually planned the entire lineup and talks, and we're really excited to get this session going. And on behalf of AST, I'd like to thank Veloxis for their continued support of CEoT. Our brilliant moderators today are Dr. Nicole Turgeon and Dr. Loy Broom. Dr. Turgeon's from University of Texas, and Dr. Bloom is from University of Pennsylvania. And I'd like to welcome our first speaker, Dr. Mandy Ford, brilliant researcher, friend, AST board member from Emory University School of Medicine. Thanks, Mandy.
All right, so thanks to the organizers, Vanita et al., for asking me to speak today. And so I've been asked to talk a little bit about kind of the basic biology of co-stimulation and co-inhibitory pathways in the activation of alloimmune responses, and kind of think about why we might want to target this class of molecules for immunosuppression and transplantation. All right. So just to get us all on the same page, of course, I don't have to tell this audience that T cells are really the orchestrators and effectors of allograft rejection. Do I have a pointer at all? No, okay. Okay. So following donor organ procurement and transplantation, there is, of course, ischemia reperfusion injury that really leads to the elaboration of molecules that serve as danger signals to the immune system. And these can be either microbial contaminants or host-derived molecules that signal damage to the tissue. These bind to damage-associated molecular pattern receptors on the surface of antigen-presenting cells that really transmit a signal to turn on that antigen-presenting cell and make it become activated, upregulate MHC molecules, and go to the secondary lymphoid organs where it can prime alloreactive T cell responses. And so during that critical event in the secondary lymphoid organs, alloreactive T cells are induced to proliferate and to differentiate into competent effectors that can then migrate back to the allograft where they mediate damage of the graft. And so it's really at that point, during that critical step of activation and differentiation, that we want to try to intervene and modulate T cell activation in order to better inhibit alloreactive T cell responses. And so the first step, of course, in alloreactive T cell activation is the perception of foreign, right? The perception of an antigenic signal. And so there are three mechanisms by which T cells can see antigen. Of course, the direct and indirect pathways have been well described over the years. The semi-direct pathway has been more well elucidated over probably the last decade or so. And this is an instance in which donor-derived MHC molecules are shed from the graft, from cells of the graft, as intact MHC and are transported on microvesicles to recipient-derived antigen-presenting cells that then can take these intact MHC molecules up and present the donor-derived peptide MHC molecules to both CD4 and CD8 positive T cells that can go on to become activated. So this is signal one. But signal one is not enough. We need also signal two. T cells need to perceive signal 2 or co-stimulation in order to become fully activated. And it's really the fine balance of co-stimulatory and co-inhibitory signals that are negative signals that function to fine-tune the response and really determine the outcome of that activating event. So you can see here, I've listed just a number of the different co-stimulatory, as indicated by the green symbol, and co-inhibitory pathways, as indicated by the red symbol, that can be at play during this interaction. So co-stimulatory signals, of course, serve to augment the proliferation, differentiation, cytokine production, cytotoxic function, memory T cell formation, and memory T cell survival. And these are counterbalanced by co-inhibitory signals that if these are engaged, they lead to cell cycle inhibition, the inhibition of effector function, tolerance, exhaustion, and apoptosis. And one of the points that I want to make about the use of co-stimulation as a class of molecules that we can think about targeting for immunosuppression is that these targets are really mostly confined to cells of the immune system. And this is in contrast to some of the targets that are used in conventional immunosuppression like calcineurin and mTOR that are very ubiquitously expressed or in multiple different cell types in multiple organs and organ systems, and therefore modulating these pathways can have more off-target effects. And so the immune modulation of co-stimulation may be more on target and have less off-target toxicities. So just for the rest of the time, I'm going to go through two examples of co-stimulation pathways that are sort of critical for T cell activation and have been well studied in the field over the years. The first is the CD28 pathway, which is an extremely critical molecule that is required for T cell activation. It's expressed on all naive T cells. And you can see here that upon ligation to its binding partners, CD80 and 86, it induces the transmission of a positive co-stimulatory signal that leads to the activation of kinases and really synergizes with signals perceived through the T cell receptor following ligation of peptide MHC that lead to the activation of NFAT and NF-kappa B and the transcription of numerous genes that sort of get the cell turned on and activated. And so CD28, like I said, is a positive signaling molecule, but it competes for the same set of shared ligands with a homologue of CD28, CTLA4. So CTLA4 also binds to CD80 and 86, but when it does so, it transmits a negative signal within the cell. And that's just sort of exemplified here, in which you can see that when CD28 binds to CD80 and 86, it leads to the activation of intracellular signaling cascades that lead to the upregulation of IL-2, increased stability of IL-2 mRNA, and of course IL-2 is a critical growth factor that is required for T cell proliferation. In this way, CD28 signaling leads to the prevention of anergy and increased cell survival. Instead, if the cell expresses CTLA4 and binds to CD80 and 86 via its CTLA4 molecule, it leads to the activation of phosphatases. Now, these phosphatases take away the phosphate groups on kinases, leading to decreased IL-2 production, increased apoptosis, and increased anergy. And so in this way, CTLA4 can really function in a cell-intrinsic manner to turn off those activating cascades and lead to anergy, apoptosis, and dampening of the immune response. In addition to this cell-intrinsic or intracellular function of CTLA4, CTLA4 also has an additional cell-extrinsic function. And this is shown here in that it is a critical for the function of regulatory T cells. So regulatory T cells are suppressive cells that constitutively express CTLA4, and when this CTLA4 binds to CD80 and 86 on the surface of an antigen-presenting cell, it can actually steal away those ligands off of that antigen-presenting cell in a process called transendocytosis. And so the upshot of this is that that antigen-presenting cell is now left with a dearth of CD80 and 86 ligands, such that when an effector cell comes along, there's no more CD80 and 86 to bind to and to activate through the CD28 pathway. So in this way, CTLA4 functions in both a cell-intrinsic and extrinsic way to critically downregulate the immune response. So to summarize this part, we know that signaling through CD28 leads to the proliferation, differentiation, and effector function. So of course the implications are if you can block that, you can better inhibit the alloimmune response. But importantly, ligating CTLA4 also sends a negative regulatory signal and allows the activation of regulatory T cell populations to also inhibit alloreactive T cell function. And if that wasn't sort of complicated enough, there are, in fact, additional complexities of the CD28 co-stimulatory pathway, which is that I told you that CD28 binds to CD80 and 86. In more recent years, it has been realized or recognized that CD28 can actually also bind to ICOS ligand, which is another ligand that can provide this activating signal. And so if CD28 doesn't have access to CD80 and 86, it can actually use this ancillary pathway to also become activated. In addition, CD80 can bind to a negative regulator, PDL1, and inhibit alloreactive T cells. So really the upshot of, or sort of takeaway message of all of this is that there are a lot of complexities, promiscuity in terms of multiple binding partners between these receptor-ligand interactions, and really understanding the mechanisms of how these molecules interact, on what cells they're expressed, and is really necessary to provide rationale for the development of immunosuppressive therapies. Okay, so the second example I'm going to touch on is the CD40, CD154 pathway. We know that CD154 is expressed on CD4 positive helper T cells, and that ligation of CD154 with its receptor CD40 leads to licensing of antigen-presenting cells, insofar as that these cells then upregulate class 1 and class 2 MHC, they upregulate costimulatory molecules and cytokines, and can better prime alloreactive effectors. And so this pathway has been well studied for a number of years in transplantation, of course, with attempts to block both the 154 and the CD40 side of the pathway. Again, touching on the theme of complexities of these co-stimulatory pathways in the immune system is the fact that in addition to its ability to bind CD40, it has recently been recognized that CD154 can actually also bind to CD11B, an integrin molecule, and perhaps bind even other integrins, such that when thinking about blocking this pathway, it's important to keep in mind this additional potential interaction. So we think that traditional CD154 binding to CD40 results in dendritic cell licensing in the lymph node. Now, like I said, that's been known for a number of years. We found that CD154-CD11B interactions actually had no effect on the priming of alloreactive T cells in the lymph node. Instead, we found that CD154 interactions with CD11B actually function locally within the allograft to increase innate immune infiltration, such that they upregulate costimulatory molecules, cytokines, overall inflammation, and can draw in innate immune cells as well as effector cells into the allograft. In addition, it's been recognized that CD154 can be expressed on endothelial cells. And so you can see here that there's the potential for CD154 expressed on the endothelium to bind to CD11B expressed on leukocytes and promote this leukocyte migration across endothelium into tissues that are inflamed, such as rejecting allograft. So again, there are implications for therapeutic targeting of this pathway in that if you're just blocking the CD154 side, you are potentially leaving open, well, if you're blocking the CD154 side, you're blocking its ability to interact with all of its receptors. In contrast, if you're blocking only one of those receptors, there's the potential for CD154 to interact with other molecules within the immune system. So again, important takeaways are that co-stimulatory pathways in the immune system have multiple binding partners, and this really provides for the checks and balances that function to fine-tune the response. But it also, this promiscuity or redundancy also adds complexity when thinking about the rational design of immunotherapeutics. And so again, it's important to understand on what cell types are these expressed, when are they expressed, when we're thinking about the design of immunotherapeutics for transplantation. Okay, and in 1 minute, I will just highlight another challenge that I think, a challenge of the use of co-stimulation in transplantation, which is really just the heterogeneity among T-cell responses. And so you can see here in the Tisney plot on the top right that if you look at any T-cell response, it's not just one homogeneous population of T cells, but rather they can be segregated into multiple distinct populations that have different gene expression profiles and are probably more or less susceptible to different routes of immunosuppression. An example of that is shown here, and going back to our CD28 example, that naive and central memory cells express high levels of CD28. But now if you look at effector and effector memory cells, you can see that there is a subset of cells that lose expression of CD28. And so if an individual had a population of alloreactive T cells that were present in that population of cells that lost CD28, you might consider whether CD28 would be effective at controlling those cells. And so I think it's important to understand then in these other populations of cells, what are the molecules that may be at play in governing the responses of these, in this case, effector and effector RA T cells. And so there are many of these. Many have been studied by a number of groups and I think are interesting for further study in transplantation. I'll call your attention to one on the bottom there, TIGIT. So, and again, in this example, you can see that within these CD28 null cells, again, that lose expression of CD28, they upregulate, if you look on the bottom panels there, they upregulate the expression of this TIGIT co-inhibitory molecule. So what is TIGIT? Like I said, it's a co-inhibitory molecule. It's actually a CD28 family member. It binds to two ligands, CD155 and CD112, again, that are a shared set of ligands for which it competes with a co-stimulatory homolog, DNAM, in a manner that is very analogous to the CD28 and CTLA4 paradigm. So you have these two homologs, one is positive, one is negative, they compete for the same shared set of ligands, and the balance of kind of who gets the signals is really important for determining the outcome of the alloimmune response. And so, again, the highlight, or I think theme here, is that heterogeneity within the immune system results in different co-stimulatory pathways being expressed on different T cell subsets, and that understanding the characteristics of alloreactive T cells within a given patient might allow for the rational design of personalized medicine to target alloreactive T cells in transplantation. I'll stop there.
Should I just keep getting? There we go. Perfect. Okay, thank you very much. Again, thank you to the organizers to putting this on. Keep co-stimulation in the minds of transplanters. These are the disclosures that are relevant to this talk. So as you heard from Dr. Ford, one of the key takeaways is a T-cell receptor interaction is just not enough. You need more to not only start an immune response, but also to determine its direction. And the CD28 molecule, clearly an important targetable molecule, but it has to be considered in the context of its co-inhibitory molecule, CD152. And all of the molecules that are up and targetable right now have this same sort of characteristic of the ligand doing something different than the receptor does, and they're being balanced between co-inhibitory and co-stimulatory receptors. The only pathway that I'll flag here as a targetable pathway of interest that wasn't mentioned in the previous talk is the PD-1, PD-L1 pathway, which is really being successfully targeted quite a bit in the oncology literature, but does play a role in how cells move through their maturation process and play a role as to whether they can be eliminated through activation-induced cell death. There's another set of receptors, CD154 and CD40, which is really active in the targeting milieu right now in transplantation. One of the interesting features of this, though, is that CD154 exists not just on cell surfaces, but as circulating trimers, particularly as they are released from platelets. So inside alpha granules of platelets, CD40 ligand trimers exist. When the platelet degranulates, it sheds CD154 around areas of trauma, and this adds another nuance to the co-inhibitory, co-stimulatory dichotomy, but also changes the dynamic of when co-stimulatory molecules exist. They exist at higher concentrations locally during trauma than they would systemically in situations where there is no trauma. Indeed, if you build a mouse that has CD154 only on its cells or only on its platelets or only on its organ, you can sort out, in fact, that platelet-derived CD154 is completely sufficient to lead to allograft rejection in the absence of any other sources of CD154. Not to say the other sources are not super important, but in fact, this is an important source of it. And if you're looking at time from transplant and the role of CD154, this is an experiment where you dope in CD154 expressing platelets or trimers. Of course, trimers will go everywhere because they're not targeted to anything. Platelets will only go to a place where there's trauma. And if you wait 30 days after a transplant and you put in trimers, you will still get rejection, but if you put in platelets, you don't get any rejection. So this points out that co-stimulation is not only a co-inhibitory and co-stimulatory pathway, but there are temporal relationships between when you see the co-stimulation and when the allograft actually occurs, which likely means that co-stimulatory molecule blockade will be of different importance right at the time of transplant than it will be long term. So as Dr. Ford pointed out, there are lots of other sources of CD154 ligation. The one that she mentioned is the CD11B, which is also called alpha M beta 2 or CR3 or MAC1. This is why it's so complicated to follow this, because each molecule has 4 different names. But there are other integrin molecules that bind to CD154, one of which is an exclusive integrin on platelets, which is quite important for surgical implications of blockade. One of them is actually embedded in the extracellular matrix of the organ itself, such that you can develop some level of co-stimulation on CD40 expressing macrophages that are migrating through the interstitial matrix. And of course, the MAC1 molecule, or CD11B, is involved in adhesion, complement activation, and coagulation. So you can see how this co-stimulatory molecule is very intimately linked to many things that are important in surgical applications. As Dr. Ford pointed out, integrin molecules add the additional literal twist in that they exist in a bent-over form that's not particularly available, but when the cell becomes activated, it actually flips up into a conformation that is easy to see. So there are changes in molecular conformation that add to the complexity of blocking these pathways, such that there are multiple co-stimulatory receptors on a variety of different cell types, and in fact, even circulating in the periphery at certain times after transplant. So as we all know, if you block combined CD28 and CD154 pathways, it's long been known that this is an exceptionally good way at preventing allograft rejection. And interestingly, if you block the T cell receptor with a calcineurin inhibitor, it makes the effectiveness of co-stimulation blockade less effective, which has made it difficult to translate these molecules into the clinic because, of course, almost everybody gets a calcineurin inhibitor. This data works not just in mice, but in primates, and indeed in the primate, the CD154 pathway is much more powerful than the CD28 B7 pathway, at least when you're blocking the B7 side of things. But both of them are really quite effective. If you look at the use of co-stimulatory blockade in non-human primate models combined with steroids, combined with mycophenolate, or combined with calcineurin inhibitors, all of the results preclinically are worse than not using those things, which is sad because every trial that has been started with a co-stimulatory molecule has used steroids and mycophenolate or a calcineurin inhibitor, where there's really no data at all supporting that methodology. mTOR inhibitors, on the other hand, are synergistic with most co-stimulatory pathway blockers. Co-stimulation permits cell division, and with it allows for activation-induced cell death. And rather than preventing cell activation like a calcineurin inhibitor, mTOR inhibitors actually facilitate activation-induced cell death. And so when you look in comparing the expression of molecules that help induce cell death of activated cells, like the PD-1 pathway, mTOR inhibitors facilitate that expression, calcineurin inhibitors prevent it. Indeed, the best non-human primate data has been obtained by inhibiting anti-CD154 with sirolimus and not blocking antigen recognition, but accentuating antigen recognition with donor-specific transfusion. So as you're looking of things to block, if you're looking to block things in the signal 2 domain, co-stimulatory, you want to actually accelerate or at least leave alone signal 1, the T cell receptor, and you want to block signal 3, which are the downstream AICD pathways. Co-stimulation blockade works here, calcineurin inhibitors block there, mTOR inhibitors block there, and mycophenolate mofetil is largely agnostic to these pathways. Increases morbidity maybe, but doesn't synergize with the pathways that we're trying to block. So the only co-stimulatory blocker that has gotten through all of the hurdles to be used in the clinic is belatacet. It was studied initially with mycophenolate and prednisone and was shown to be a really good replacement for calcineurin inhibitors if you could get through the early phase of acute rejection. In all the experience with blocking the B7 side of this receptor ligand area, there's much more early rejection, but once you get through that, patients do quite well. Most of this early rejection is been associated with various forms of memory or matured T cells. As Dr. Ford pointed out, T cells are not a homogeneous thing, but there's multiple different flavors, all of which have different dependencies on this pathway. And it's important to note that as you start picking up maturation for T cells and losing dependence on co-stimulation, you actually start gaining adhesion molecules that drive cells toward sites of inflammation. So most of the problems with early rejection with belatacet can be mapped to various characteristics of these memory type T cells. Interestingly, if you treat someone with a calcineurin inhibitor and you follow their prevalence of memory cells, it actually never changes. CNIs prevent the loss in the AICD of memory, and therefore, when you come off the calcineurin inhibitors, you're likely to have rebound rejection because you haven't really gotten rid of the cells that you needed the CNIs for. You also, when you deplete T cells, they have a different propensity to grow back. So using depleting agents with co-stimulation blockade actually provides a requirement for IL-7 receptors, and memory T cells don't have a lot of IL-7 receptors, so naive co-stimulatory sensitive cells can actually outcompete them and make it a more prominent cell type. Indeed, if in this study that was done with belatacept, you could follow the gaining of CD28 positive naive cells and the loss of CD28 negative memory cells over time just by going through the process of cell depletion and repopulation. In this trial, we were able to get people, about half the people that we tried to wean, to just being on belatacept, which was proof of concept that co-stimulation blockade, once you get through all the instability of the transplant period, can be completely and solely capable of preventing rejection. And our longest follow-up in this trial now is over 10 years, where there are people that are just taking every other month belatacept as their only drug for preventing rejection. If you can get off of calcineurin inhibitors and other immunosuppressants, it's been now shown that renal function gets better. Our thought that all renal patients have to slowly have a demise of their kidney function is a product of our dependence on calcineurin inhibitors. And if you don't depend on calcineurin inhibitors, you can move to calcineurin-free environments, you actually get better function over time. So now talking about blocking the other side of belatacept, CD28, there was an attempt at this about 20 years ago, which used an agonist to CD28, which was catastrophic and led to cytokine storm and serious illness in patients that got this. So a lot of work had to be done to look at non-agonist CD28 ligands to go into the clinic. This is a study from about 10 or so years ago looking at FR104, which is a CD28 specific DAB, which was shown in this paper not to be stimulatory, and in the non-human primate work that came from this showed that it was actually quite effective in preventing rejection. This drug has now gone on to the clinic to be what's called VEL-101, which is getting ready for clinical trials based on clinicaltrials.gov, and will be the first, will be one of the first CD28 specific agents to get into the clinic to test this hypothesis that if you bind CD28 and not B7, it will be more effective. There is another CD28 specific molecule that's out there, lulizumab. This has been preparing for clinical trials, at least according to clinicaltrials.gov, for almost 2 years. We're sort of waiting to see where that's going to happen. There was a small experience with lulizumab in humans that was in a trial with that was run by Flavio Vincenti, but enrollment was halted in that trial based on concerns that efficacy, at least for this regimen, was not particularly good. Very small experience, though. Looking at anti-CD40 ligand, the first molecules out of the chutes and in the clinic block CD40. This was before people really had a good understanding of the fact that CD40 blockade is very different than CD40 ligand blockade. This study with blasilumab showed actually pretty high rejection rates, and that trial was stopped. Iscalimab also looked like a really promising CD40 inhibitor, but very recently this trial was discontinued, and the data, not published in peer review but now available on clinicaltrials.gov, suggested not so much that the molecule was not efficacious, but that there was enhanced toxicity to the arm that got this drug for reasons that are not really clear. So we'll have to wait for those data to be come out in the peer-reviewed literature. There is another CD40 specific molecule, KPL-404, that's in trials in rheumatoid arthritis. But more interesting to the transplant literature is this was the drug that was used in the first cardiac xenotransplant, which was remarkably successful despite the fact that the patient ultimately died about 2 months afterwards. Finally, there's a CD40 ligand molecule out there now that is attached to human serum albumin instead of being an antibody or a typical fusion protein. And there is a trial that really went back to the original approach of blocking CD40 ligand and blocking the B7 CD28 pathway with no other immunosuppressants. This trial has just finished enrollment. Hopefully the results will be presented at the ATC this year. But in the preliminary look at the data, it looks like using co-stimulatory blockade the way it was originally discovered, instead of trying to shoehorn it into current standards of care, may actually be a good way of going forward. Unfortunately, this drug has been bought by 2 different companies since the original trial got started, so we are sort of at the mercy of whatever the most recent company is, currently Amgen. Finally, there is a anti-CD40 ligand antibody that has been modified on its Fc receptor. It's made by Eledon, and this has given really promising results in non-human primates. Doesn't appear to have activating effects with platelets, and hopefully will be going into clinical trials in the next year or so as well. So to summarize, there are innumerable targetable co-stimulation pathways. Many have variable clinical agents and are in clinical trials. They direct, but they don't initiate immune responses. You still need the T cell receptor to initiate the immune response. Targeting the receptor can have widely different effects than targeting the ligand. The effects of B7 related pathways are more T cell specific, including T cell dependent B cell activation, and more impactful for new immune encounters instead of memory encounters. The CD154 related pathways have broader effects, including modulating the impact of trauma and immune activation. And based on preclinical data, the effects of B7 and CD154 blockade are synergistic. They are antagonized by calcineurin inhibitors. They are synergized by mTOR inhibitors, and they're largely unrelated to antimetabolites. It appears that depletional induction will be important in controlling early rejection with any of these agents. And I'll stop there. Thanks very much.
Lessons learned from the first generation co-stimulatory blockade and implications for clinical trial endpoints.
Well, good afternoon. Thanks very much. I want to thank the organizers for the invitation, and obviously thank them for allowing me to follow Dr. Kirk and Dr. Ford, which, of course, is never an easy act to follow. So happy to be included here. I'm going to try to bring this around to talking about clinical endpoints, which I think is an exceedingly important topic for us right now. These are my disclosures. That slide's needs to be updated. There's a lot. I'm busy, obviously. My objectives that were given to me were really to enumerate reasons for new endpoints for clinical trials with co-stimulation blockade. And I thought where I'd start is looking at why we're interested in new molecules. And this is all obvious to us, is that the limitations of CNIs, which are not really counted by regulatory authorities at this point in time. These are things that matter to me and my patients: diabetes, suboptimal kidney function, cardiovascular risk factors, cosmetic changes, and neurocognitive changes. And these are obviously not an exhaustive list. This was my sort of top 5 things that I don't like about CNIs that don't matter from a regulatory perspective. I think I'd start by saying let's go back to the belatacept experience and just provide a scorecard of what we've learned from this experience in terms of clinical outcomes. So Not meaning to be exhaustive, I'm just focusing on the benefit trial here, which I think is familiar to everybody in this room. And really what was, I think, really striking is the 7-year improvement in graft survival. And I'm gonna really harp on that in a second here, because really that's a remarkable achievement. Of course, this is in comparison to cyclosporin, as everybody here knows. But when you look at and this was, of course, despite the fact that there was an increase in rejection with the use of these drugs that Alan has already covered. When you look at the actual survival curves, you can see the total graft survival differences, and they're statistically significant on my left, which would be your right, for total graft survival. The individual outcomes were not statistically significant, but you can clearly see over the long term that there's separation of these curves. Now, importantly, one of the criticisms of the long-term data is that at 7 years, you had more attrition in the cyclosporin arm, and that's true. I think you can see that the drop-off, if you can read that, those numbers along the bottom, is higher in the cyclosporin group. But I still think this is a remarkable clinical trial with 7 years of follow-up, showing an improvement in an outcome that even the regulators can agree is important. What about the other outcomes? The first is eGFR, and I'm a nephrologist, so I'm going to speak a fair amount about that. I want to touch on the issue with EBV and PTLD. We know that this is confined to EBV naive patients. One of the things that I think is a takeaway from perhaps Mandy's talk is the fact that, you know, co-stimulation has all these promiscuous effects. And for me as a clinician, as we think about managing viral infections with these drugs, one of the things that we need to know how to do is to dial these drugs down when we have iatrogenica imperfecta. And I think that's perhaps a discussion point for our discussion as we go forward. Importantly, a decrease in donor-specific antibody formation, which I don't think either Alan or Mandy highlighted the mechanisms that underlie that, but that could be something that we talk about in the discussion. And importantly, I put a number of question marks up there. That's not because the designers or the architects of these trials were lazy. It's because our clinical endpoints that are relevant in the clinic change over time. So these are things that I'm sure matter to Dr. Cohen and myself, BK infections, diabetes, proteinuria, but these are not things that were counted or reported in the primary control trial data. Of course, there's been observational study from port investigators, for example, suggesting that cardiovascular risk factors might be more profiled, but from the control trials, these things were not reported out. Now, Alan showed this slide, which is the eGFR changes, and I'm just going to sort of take you through this from how a nephrologist thinks about it. The first early separation that you see with the two belatacept arms and the calcineurin inhibitors is obviously a hemodynamic effect, right? Stuff hasn't been happening long enough for there to be a meaningful difference in damage. But over time, as Alan pointed out, there's a clear, and I think everybody would agree that that's a clinically significant difference in GFR. Now, some people have pointed out that it's implausible that GFR would increase over time in the belatacept treated groups, and they wonder about hyperfiltration. And of course, as I showed you on the previous slide, proteinuria wasn't measured. I think that is an important issue, but I would say irrespective of mechanism, this is an impressive difference in kidney function. We'll come back to GFR more. This is what I would care about as a clinician. So this was included in the supplementary of the original trial. It shows you probability of survival, graft loss, and eGFR less than 30. And this is really impressive compared to cyclosporin. So I could pause here and saying if cyclosporin was all we've had, you know, we wouldn't need new trial endpoints. This is pretty compelling long-term data that really shows remarkable success with the use of co-stimulation blockade. Of course, we're using tacrolimus as a mainstream, and that was, of course, mandated by the regulatory authorities at the time the trial was done. I'm not going to take you through the limited control data. There's 4 trials. If you care to look them up, they're in that publication that I show you at the better, but we're talking small numbers of patients in controlled trials using tacrolimus compared to belatacept. So this brings us now to the important discussion of surrogate endpoints, and I think that we all recognize that to advance new therapeutics, whether they be co-stimulatory blockade or not, we are going to need the advancement of surrogate endpoints that are meaningful to our patients, but also obviously accepted by regulators. And I'm going to start with the basics because, we think we know a lot about surrogates, but revisiting the Prentice criteria, which are really a high bar for any surrogate to make. And the big issue here is that an ideal surrogate is one that is on the only causal pathway for the disease process. And the intervention, whatever it may be, is entirely encapsulated by its effect on the surrogate. And that's something really important for us to get a handle on at what a high bar that is to achieve in these criteria. This slide is meant to show you why various surrogates will fail. And again, it's all based on the Prentice criteria. In the top picture, you can see that the surrogate is just not on the disease pathway, so it's just out in left field. In the second instance, there's many pathways to the disease, but the treatment only affects the pathway through the surrogate. In the third case, the surrogate is not on the pathway of the treatment effect, or it's insensitive to the treatment effect. And in the last case, in panel D, the treatment acts through mechanisms that are independent of the disease process involving the surrogate. And this is, I think, this basic understanding of surrogates is really important for us to think about as we think about new therapeutic endpoints. I'm just going to bring this home using the Symphony example as a trial we're all familiar with. This was a huge trial, obviously, and we all know that the low-dose TAC arm had better GFR and it had better survival. And so the question is could GFR in this trial act as a surrogate? And the issue, of course, was that the low-dose TAC arm also had lower rejection rates. So if we put this back into the framework that I showed you previously, really there's a question about whether the effect on long-term survival that you saw with the low-dose TAC arm is mediated through GFR, or is it mediated through acute rejection, a little of both, and would it be encapsulated by GFR, the acute rejection effect? So that just shows you the complexity of trying to nail this down according to the Prentice criteria. Now, the reason this, of course, is important is for us to advance new therapeutics, we need to really go for expedited drug approval. And the most important thing is in the first bullet point here is that the intention here is to facilitate development of new drugs to address unmet needs for the treatment of serious or life-threatening conditions. And I think one of the battles that we have currently with our regulatory bodies is I'm not sure that they believe that the needs of our kidney transplant candidates fall into that category of a serious disease in need of accelerated approval. We're all fixated on whether our surrogates are good enough, but I think part of the question really is are they accepting that first premise? And this, of course, brings us up to a definition that I think we should all be familiar with, which is this concept of a reasonably likely surrogate. I think we've talked about it, what a validated surrogate is. This is something that could substitute for a hard outcome, but a reasonably likely surrogate is one for which you have biological plausibility, you have epidemiological data, and it's something that can serve as a surrogate, and then of course you still have to do the long-term follow-up to show that it marries with the long-term outcomes. But if we don't get the reasonably likely surrogates in the gates, we'll never do the trials to prove that second component of it, and that's why this is so important. This is just sort of a scorecard from Martin Nasons about the status of current surrogates. And so basically you can see the criteria listed on the left, or that'd be your right. And then you can see that the I box is which is a composite endpoint, is the farthest along in terms of being confirmed as a reasonably as a surrogate outcome. I'm going to come back and talk about GFR with or without proteinuria a little bit here in the last couple of minutes, because as we all know, in the clazakizumab trial, this has been advanced as a reasonably likely surrogate in that trial. So there still is significant interest in kidney function itself. Now, again, I'm a nephrologist, so I'm going to harp on kidney function. And just to review, why is GFR not work? We're all familiar with that Harry Heron paper at 1 year that shows GFR is good, and that obviously associates with long-term graft survival. So why isn't that good enough? And the real issue is these injury processes that contribute to late graft failure are maybe subclinical, and not captured by an early decline in kidney function. So if you're talking about a single point endpoint, that could be an issue, or you could have compensatory hyperfiltration, which really covers that up. And for that reason, as pointed out by Bruce Kaplan and Ulf and others, this alone as a single endpoint doesn't really hold up as a surrogate. But eGFR slope might be different than that, and we've seen in chronic kidney disease that this is being embraced, and in fact they've made great progress, and we're behind them where they are at this. I would point out that there are some challenges for co-stimulatory blockade that we have to address if we're really going to seriously talk about eGFR slope, and that is the hemodynamic effect. And we're all familiar with this in the context of ACE inhibitors, or more recently SGLT2 inhibitors, and the way they get around it in native kidney disease is they basically have a baseline, which is a challenge for us because in de novo trials we don't have that, and then they take you off the drug at the end of the time and they can sort out what is the hemodynamic effect versus not. That's going to be a challenge for us because obviously we can't really pull immunosuppression, but I think hammering out exactly what the hemodynamic effect of these drugs is, if we're going to be comparing them to small molecules like calcineurin inhibitors, is going to be an important component, whether we're talking about kidney function as a primary outcome or a secondary outcome. In the last couple of minutes here, I want to sort of blow your mind about thinking about something a little bit differently. So we know that drugs can be approved by how people feel and function, and not just survive. And this is really part of the FDA's not new, but initiative on patient-focused drug development. I'm going to put up the top quote by Janet Woodcock, who was the commissioner of the FDA, and I like this quote. It says, "It turns out what's really bothering your patient is different from what's bothering the doctor." And so really the issue is having our drugs meet patient needs with regards to this. And there's many ways that we can get these outcome assessments about how a patient feels or functions, but I can tell you that what determines when I see Mr. Smith in the clinic on Monday what the rest of his week is going to be like and how he feels about himself is what I tell him his kidney function is going to be like. Now, that's a bit of a humorous example, but in truth, there is data that chronic kidney disease stage marries with health-reported outcomes reported by patients that are clinically meaningful. And more importantly, we know that kidney function definitely marries with resource requirement, like things like hospitalization and other sources of morbidity. And I think this is an important issue. So thinking about kidney function not as a predictive outcome, but more as an outcome that actually is a metric of how people feel or function is something that I think we should start talking about, because I think there is a pathway, which is shown here, to how to get these patient-measured outcomes accelerated to approval. Now, this is a long process. It means that we have to start talking about qualitative things, which none of us like, because let's face it, to the qualitative researchers in the room, it's a black box and it needs to be much more tighter. But if we could marry it to something that we can all hang our hat on, like GFR, and say these things matter in terms of how a patient functions, I think that's something that we should discuss. This is my last two slides. I'm going to wrap up here. This is provided to me by Bill Fitzsimmons. And really what Bill's done here is really categorize various outcomes in terms of accepted primary efficacy outcomes, secondary. And the third box, I think, is really important. These are what I would call safety outcomes, which have been accepted as primary outcomes in other disease states. And the point here is that there may be wiggle room to have these outcomes elevated to where they may be accepted by regulators. And the last column is obviously the patient-reported outcomes that we alluded to earlier. What I've done here is color the ones that I think will potentially be better off in a co-stimulatory environment. And I think it's us to think about which are the leading contenders that can either be accepted as primary or secondary outcomes by regulatory authorities. I'll end there. Here's my summary. So new regulatory approvals are dependent on acceptance of reasonably likely surrogates. The iBox is currently the leading contender. I don't think kidney function is dead, and I think that we should examine that more closely. I think that we should potentially think about kidney function not as a predictive surrogate, but a biomarker of how people feel and function. And I think most importantly, the regulators must acknowledge, and this is a battle that I think we're waging right now, that transplantation is a serious condition with vast unmet patient needs. And to that end, I couldn't help but put in a plug-in for the AST patient survey, which will be coming your way at ATC. Thanks very much.
thank our distinguished panel of speakers and invite our speakers to come up here so that we can have a panel discussion. And we also invite our audience to ask questions. It's not every day you get an opportunity to have this panel of speakers available to you, so please take advantage of that and ask some interesting questions. No takers.
Okay, I'll get the ball rolling. You had a question? Go ahead.
So my question is for Dr. Kirk. So, your slides about platelet activation are interesting, and one of the things that I've always, or that I think has come out in the literature as being really important, an important effect of the anti-CD154 molecules, reagents, is their propensity to lead to regulatory T-cell differentiation. And it occurred to me that platelets, when they get activated, secrete a lot of TGF-beta, or to your knowledge, has that been looked at as a source of how the anti-CD154 leads to Treg induction, knowing that obviously TGF-beta is an important cytokine that differentiates cells into Tregs?
So, no, to my knowledge, no one has looked at platelet-derived TGF-beta as a etiological source of Treg development, but someone probably will after this meeting. So yeah, maybe you should go do that, right? Yeah.
Is there any, so at some point when co-stimulation first became sort of an entity, there was enthusiasm at looking at antisense therapies. What has ever happened to sort of that line of investigation? And is there any reason that there hasn't been any progress?
There was one small trial with an antisense molecule looking at reperfusion injury that had negative results and didn't go anywhere. It wasn't co-stimulation specific, but it was an attempt to get that mechanism of drug delivery into the world. But since then, I don't know of any company that has tried to use that as a mechanism. The delivery of antisense therapy is tricky, and getting it to where you want it to go also really tricky. And a lot of the co-stimulatory molecule stuff, particularly CD154, is secretion of existing created protein. So if you block transcription, it's not going to do anything to what's like balled up in platelets or sitting in dendritic cell vesicles or things like that. And that might be, at least for an acute issue, a problem with antisense therapy.
I got a question for Alan, actually. Alan, you really brought out the time course of mechanisms of action and how they differ. And from a clinical perspective, obviously the one we face is getting over the hurdle of rejection. You mentioned ischemic reperfusion injury, but the Australian data would suggest that in the long term, malignancy is something that we have not paid enough attention to. Can you speculate about the time course and potential long-term advantages from a malignancy perspective? Obviously, the DSA stuff is stuff we love. We love the GFR stuff, but what about cancer?
So a lot of the Australian literature is heavily skewed by skin cancers, and so you have to sort of strip that away from the data to see how much malignancy issue there really is. But my sense is that virally driven malignancies will be a problem with any of the immunosuppressive regimens that we have, including co-stimulation blockade. And for co-stimulation blockade, particularly viral malignancies that are driven by newly acquired viruses after you're on therapy. So if you've picked up the viruses that you're going to pick up and then you get put on a co-stimulatory inhibitor, probably is not going to do much. But for example, EBV naive people that get an EBV positive graft, we know that pushes lymphoma limits from Epstein-Barr driven PTLD. For the non-virally driven malignancies, Yeah, I don't know that it's gonna be, that much of a problem, just because of the redundancy. I mean, you're blocking one co-stimulatory pathway, but, there's a lot of redundancy, as Mandy pointed out. And of course, you know, whatever you're blocking, malignancies find their way around it. That's part of it, so. That's the way I put it together. Anyway.
Ironically, some of the malignancies you'd be treating with immune checkpoint inhibitors.
So, yeah, that's actually a really interesting question. We've had some people that are on Veloxis that have then gotten a malignancy and gotten treated with an immune checkpoint inhibitor, and remarkably, nothing happened. What you thought, you would think, oh, that's going to screw everything up.
Nothing happened to the transplant.
So the transplant, but the cancer got fixed. Wow. So it's way more complicated than any of the, you know, cartoons that we put up as to how this comes together. I do think that it'll be interesting to come up with a biologic that can be reversible too. One of the problems with a biologic is you're on it and, you know, you've got at least 2 months of tail from the time you dose it. To come up with a way of reversing the effects of a biologic when you get into trouble, I think would help people.
Yeah, I think that's a really important issue because you pointed out the importance of T-cell depletion early on. So we're all focusing on rejection, avoidance of that. And this is, I think, is a really important challenge, either whether it be spreading out the dosing, which you've shown in the long term with the spreading out the dosing in the long term, but early on, I think this is, the control of that is a key issue. Yeah.
And then to that point, can I just, since we're, obviously one of the, so if you look at the data with belatacept, you know, you can get more severe CMV and this resistant CMV and more greater susceptibility to viruses. So do you think that second generation co-stimulation blockade, this is not just for Alan, but for anyone on the panel, do you think we should be able to monitor levels? Do you think that and if we could monitor levels, do we know what that would reflect the extent of immunosuppression? But you know, before we had co-stimulation blockade, there was this whole thing, well, MMF, you can't measure levels and you don't really know what you're doing. Are we potentially leading ourselves down the same path with co-stim blockade?
I'll take that one on, because to me, one of the obvious potential advantages of co-stimulatory blockade is from an adherence perspective, right? You know they've got it on board. And so I think having a good mechanism, so whether it's receptor occupancy, actually Ulf and I were talking about this, I think over a cocktail at some point, we were, you know, if that is then something that we could use in the clinic as a mechanism to dial things up or down, I think that's an important way to measure things, Roy. So I think it's exceedingly important. Yeah.
We have a question?
Yes, thank you very much. Tom McCune from Centera Novel General, EVMS. You discussed the occurrence of non-viral associated carcinomas while on belatacept versus calcineurin inhibitors. My question to you is, and it's to the panel as well, and to the room as well, what is the best conversion immunosuppression when your patient does develop squamous cell carcinoma? Because right now I'm hearing that we always try to get them off the anti-metabolite and keep them on the calcineurin, but I'm now hearing that calcineurins may actually be upregulating the epidermal growth factor receptor. So I'm throwing this out to you as, is co-stimulatory receptor conversion and removal of the calcineurin better than keeping somebody on a calcineurin and minimizing the anti-metabolite?
Kind of depends on the kind of cancer.
Yeah, so if you're talking about squamous cell, like really aggressive squamous cells, I've had a couple of patients that have gotten them and needed like skin grafts and things like that. I've found that after they get healed from their resection, putting them on an mTOR inhibitor is a reasonable way of going, because mTOR inhibitors, particularly for hypervascular tumors, are primary therapies for they're anti-cancer drugs. That's what the company is making them for right now. And so particularly for people that are prone to squamous cell and things like that, I've gotten a lot of them transferred onto sometimes just monotherapy mTOR inhibitors. The problem with that is if they're on them and they have to have a skin graft, the wound healing is really challenging. So sometimes we'll put them on a calcineurin inhibitor just to get them through the operation and then get them switched over to an mTOR inhibitor.
Just to change gears a little bit, but question for Dr. Gill. Transplant's probably been a victim of its own success. Everyone really does well, really, certainly early on. And part of that problem is that, we're trying to improve long-term outcomes, but we're using short-term outcomes to guide, how we treat patients in the long term, right? So clinical trials are, 1 year, 2 years, and then we use that information to guide lifelong therapy. So can you expand a little bit upon, how you would change metrics, how the FDA should change metrics for us to be able to improve 10, 15, 20-year outcomes?
I think you've broadened the topic beyond this, but I'm glad you have. I think death with a functioning graft and defining when that's a premature death with a functioning graft is a really important thing that needs more discussion, right? So, a successful transplant is somebody whose graft lasts 1 day longer than they would have survived, right? So death with a functioning graft isn't always a failure. So I think really defining that, which we've had relatively little discussion on. Now, is that going to be fodder for a regulatory target in a trial? And the answer is probably not, right? Because it's a long-term process. But I think that their biological plausibility of, and we all know the shortcomings of, for example, the Framingham risk score for predicting cardiac risk, right? And so forth. But that's where I think some of the, if you could show mechanistically, for example, with malignancy, you could show an intermediate outcome that has biological plausibility, that you're changing a pathway, and Mandy talked about potentially even tailoring co-stimulatory blockade for people with different competing risks. So whether they be cancer or cardiovascular disease, for example, that would be very exciting. And I think that you would have to make the case that these are then surrogates that are going to be predictive of long-term outcomes. So that's a tall order. But I think what would be exciting, and perhaps as a differentiator from a safety or a secondary approval, is to say, look, you've got, let's say, someone who's got a history of skin cancer, and this co-stimulatory pathway is really friendly for that. I think that would be compelling. Would it ever elevate to a primary outcome? Probably not. I think that would be a tall challenge. So I didn't answer your question directly, but hopefully you can see that if we advance some of these mechanistic targets, I think that that's interesting. And just to say that that's not so pie in the sky, right? For diabetes, people are talking about hemoglobin A1C control as a surrogate for long-term complications of diabetes that might be accepted. So we have to really advocate for that, but I think it can be done.
So what's the hemoglobin A1C for transplant?
Well, yeah, we got to come up with that. Dr. Kirk and Dr. Ford will come up with that. That's outside my pay grade. Yeah.
So the direct trial actually years ago did look at that, but to just sort of taking that a step further, if you look at the potential side effect complication or side effects of therapies that could potentially be surrogate endpoints, diabetes, hypertension. Interestingly, you picked up on cardiovascular endpoints, which for obvious reasons. Assuming, and we know from observational data that like even developing new onset diabetes increases the risk of cardiovascular disease and is associated with increased mortality. So assuming that was borne out in a randomized controlled trial, patients are going to die from something, and we know that death is the leading cause of graft loss, so that comes back to malignancy. And so do you think that should also be, you didn't highlight it as a rare, as an important thing, but since we don't know, since there's a potential association with viruses, should we be elevating that?
Well, I mean, I think that the other conversation that I think has to come into this, the answer is yes, Roy, I think, to your question, but I think what we need to think about is real-world evidence for this, right? So Ilka Halantra has written about this, right? Our data, if we think about our field compared to every other field of medicine, right? We're regulated. We know when we start the disease process, i.e. the day of the transplant. We've got great data sets. We've never effectively leveraged those to provide real-world evidence, so to actually show that these differences matter. And I think we have to sort of go there, right? If we're really going to tackle long-term outcomes, We have to have much better data, including therapeutics, treatments, et cetera, and response to therapy. And that's not such a crazy order to think about, right? We just need to rethink the system.
Thanks. Hi, my name's Drew Silverman, and I'm a transplant pharmacist. So one of the things when you look at the benefit trials is the early rejections that we saw. A lot of it has to do, in my mind, with the fact that we don't know what we're doing with CELCEPT, right? And if some of what you pointed out is that early in the process, you have upregulation of adhesion molecules, right? So one of the thoughts, and it's coming across pretty clearly here, is being able to do co-stimulatory blocker and actually use MPAs and measure AUCs so that we know what we're doing with them early, and then looking at a time where you would convert from an MPA to an mTOR to prevent a lot of these long-term problems that we're discussing right now.
So I do think a lot of people have gotten into a mindset of using MPA in the first month, and then switching over to an mTOR when the wound is healed. I don't think anybody is being attentive to AUCs of MPA, which it would be easy to do, but it would be easy to do with calcineurin inhibitors too, but we don't do that either. You know, when I have somebody that is got a rejection on calcineurin inhibitor or an infection despite the fact that they have normal trough levels, if you do an AUC, 50% of them, despite a normal trough, have, you know, way out of range drug exposure. And I'm sure that it's the same for MPA. So that's a social experiment for physicians. It's not a biological experiment to get people to just use the tests that exist for them.
Although the Opticept trial showed that there was no benefit in the TAC regimen to measuring AUCs or MPA levels.
Just bringing that back to outcomes, I know we're short of time. I think that this is an exceedingly important issue, right? When we talk about surrogates, if you had agent A that provided much more, a higher level or percentage of your patients that were in therapeutic target, right? That should be an ideal surrogate, right? And so new therapeutics, so we were talking about receptor occupancy, for example, with co-stimulatory blockades, or other mechanisms that you could say that with this drug, we have a much higher achievement of therapeutic target for patients, and it's easier for the patients. That's the type of conversation we should be thinking about.
We have time for one more question, John?
Yeah, thank you. Kabashigawa in Los Angeles. We in cardiac, we're starting to use belatacept. And one of the concerns we had was the rejection. And particularly now, T-cell mediated rejection seems to be implicated more so in cardiac allograft vasculopathy. We saw that in the cardiac iBox. And in fact, you know, the T-cell mediated rejection was even more of a factor as opposed to antibody mediated rejection. Yeah, and DSA was involved as well. But what are your are we right to be concerned in terms of chronic rejection from the from having more rejection potentially?
I think the early rejections in co-stimulatory trials are very different than early rejections in calcineurin inhibitor trials. They have different consequences, much less consequence, and even though they seem to have high pathological grade, they are really steroid responsive, and it's just not the same disease. We've also seen in all of the co-stimulatory trials that if you get through that early period, late rejection is exceedingly rare, which is very different than what happens in calcineurin inhibitors. And I think that's because the pathways allow for regulation and adaptation, whereas calcineurin inhibitors just lock the immune system up, don't let it do anything bad or good. And so I think that over the long period of time, you'll have less vasculopathy if you can navigate the first 3 to 6 months.
By the same token, should we be going with a PSI as opposed to a MMF
Going with,
with everolimus, sirolimus.
I think sirolimus, if you can get out of you get the sternotomy healed, then you need, it's fine to use an mTOR inhibitor. And in fact, it's probably good for the coronaries as well. I mean, you guys are sticking sirolimus-coated stents in there, so.
Thank you for reminding us. Thanks.
I just wanted to riff off Alan's point about the difference with rejection from a co-stimulatory versus under a CNI. There's the AJT that really focused on the fact that we're not treating rejection effectively. I would say that actually those early rejections that are by co-stimulant are probably don't require even the doses of steroids. Like everybody writes, half a gram of Solumedrol for 3 days and then they taper it over 3 months, right? I think that may be too much. And I think that those are the types of things when we even examine how we treat early T-cell rejection, they're not all the same depending on what's on board.